A brain tumor is a terrifying disease. It grows inside the brain, severely impacting patients' daily lives and generally killing them within 15 months. Glioblastoma multiforme makes up 52% of all brain tumors, affecting 3 in 100,000 people, with nearly 23,000 cases per year worldwide. Standard treatment consists of surgical resection of the tumoral mass, followed by cycles of radio and chemotherapy. A cure for this disease is yet to be found. Why is glioblastoma so hard to treat? Firstly, because it is extremely aggressive. Secondly, because of its location, the brain. Any proposed therapy should halt tumor growth while guaranteeing normal brain functions and limiting damage to healthy brain cells. The main problem is that the brain is surrounded by a blood-brain barrier, a barrier that only allows for the passage of water, some gases, and minuscule molecules. This barrier is supposed to protect the brain from pathogens. Hence, any proposed therapy should be able to cross that barrier in order to get into the brain. My research has demonstrated that one specific bacterial protein, CNF1, derived from E. coli, counteracts glioma growth and preserves neuronal function when administered directly into the brain. However, CNF1 cannot cross the blood-brain barrier by itself. My proposal overcomes this limitation by fusing CNF1 with chlorotoxin, a protein produced by a North African scorpion. We know that chlorotoxin can cross the blood-brain barrier and also can selectively recognize glioma cells. Thus, chlorotoxin becomes the taxi that delivers the drug, CNF1, into the brain. Here, it will attack glioma cells, blocking tumor growth, protecting neuronal tissue, and ultimately allowing 23,000 people a year to live far longer. Thank you for your attention.
Exactly what does CNF1 do in the brain?
So in the brain, it preserves the neuronal spines. So when the tumor grows, it stops the communication between neurons, and then neurons start to die. So CNF1 can protect the architecture and the functionality of those neurons.
Have you done any preclinical trials?
Not yet. We are in the lab still. Yeah.
Thanks for the talk. How do you administer a bacterial protein into the bloodstream without getting an immune reaction against it?
You mean CNF1 or the both?
Diffusion.
Diffusion, I was thinking about endovenous treatment because it's like what happens with chemotherapy.
Thank you very much.
Thank you.
Thank you.